Cool. There we go. Yeah. Cool. So the title of my talk is Expressing Any Gene Anywhere, Anytime, and it's based around my PhD thesis. So many modern therapies rely on giving the cell new instructions in the form of genes. The issue, however, arises when those genes turn on in the wrong tissues, as they lead to toxicity and even cancer progression, which can obviously be detrimental. So in order to explain how those genes are delivered, I'm going to give you a speedy class on gene regulation. And those types of constructs, by the way, are called transgene constructs. So in the mammalian genome, expression is established at the promoter element, where the core transcriptional machinery is recruited. However, promoters are mostly unable to self-activate, and they require additional information coming from distal enhancer elements. Enhancers interact with their target promoters and provide information on when and where to express the target gene. This is why transgene constructs are typically designed by bringing enhancers and promoters into close proximity. This is unfortunately an oversimplification. Every single cell in your body contains around 2 meters of DNA that needs to get compacted and packaged into different chromosomes. And that's the reason why identifying the relationship between an enhancer and a promoter is not a trivial thing to do. This is why typical transgene constructs actually contain an enhancer providing that cell and tissue specific portion of the construct, followed by a promoter. There's a generic promoter that typically comes from a viral origin. The issue with this approach is that the construct size is relatively large, and it leads to leaky and unwanted expression in the wrong tissues, which limits the amount of dosage that can be given to a patient, and thus limits also the therapeutic benefit. This brings me to my PhD thesis, where I've been working on generating transcriptionally autonomous enhancers. What we've done here is to remove the promoter altogether and harness the specificity from the enhancer. This makes the construct significantly smaller because we don't need the promoter. There's no leaky expression and therefore no toxicity. And there are millions of cell-type specific enhancers in the human genome alone, which means that we can make as many chances as we want to do. For the biologists in the room, I'm going to explain a little bit more what transfers are. And essentially what we've figured out is that we can stabilize enhancer transcription by just adding a couple sequences. Those sequences are required to interact with the nuclear to cytoplasmic export machinery. This is because transcription happens in the nucleus, whereas protein expression takes place in the cytoplasm. And we also need to stabilize the 3' UTR of the cargo. We do this by adding a couple sequences that I've summarized in this table here. But unfortunately, based on time, I'm not going to actually go through the different elements. Since I believe that images speak louder than words, I am going to run you through a couple in vivo results. So here, what I've done is to identify a heart enhancer that I've linked with a green fluorescent protein, and we've done this experiment in zebrafish. So here, I'm just showing you a wild-type zebrafish. For someone that's not seen a zebrafish before, I've labeled it in the image. And the reason why I'm showing you a wild-type control is so you can see that in the yolk sac, we get a little bit of background fluorescence. And this is totally normal. It's based on the components of the yolk sac. However, when we introduce the transfer construct, now you can see tissue-specific expression in the heart of the zebrafish. For some people there, you can also see expression in the heart, and this is again totally normal, and it's based on the expression pattern of this enhancer that also tends to express in the eye. Given that zebrafish are not all that related to humans, we also wanted to perform some experiments in more closely related model organisms, such as mice. And so in this case, I selected a lung-specific enhancer, and I linked it with a luminescence marker. This is just a diagram to show you what the lungs are within a mouse model. And in this image, you can see the blue luminescence marker expressing in the lung of a mouse. As you can observe here, the expression is not happening in the entire lung, and this is because we're wanting to do a very nuanced expression to be only in a very specific cell type within the lung. And that cell type is the alveolar tissue, and it's because it's the one affected in diseases such as cystic fibrosis. But what are transits useful for? I've been hinting at different applications, but actually, what this technology allows us to do is to express any gene anywhere at any time, which means that it could have potential applications ranging from basic research to more translational applications. And just to finish off my presentation, I'll be focusing on how I've done proof of concept data in immuno-oncology research. So blinatumomab is a bispecific antibody that binds CD3 receptors in the cell surface of T cells and CD19 receptors in the cell surface of cancer cells. What this bispecific antibody does is it activates the immune system, the T cells that it's binding, targeting the cancer cells for degradation. Unfortunately, not all cancer cells express CD19, and they can also escape treatment by just mutating the expression of the cell surface receptor. And therefore, in the case of blinatumomab, it will still lead to cancer progression. So what I decided to do here is to find a cancer cell line that doesn't express CD19 naturally, and this is what the K562 cells are, which are a leukemia model. I then introduced a transfer that expresses in the K562 cells, and as you can see here, in the presence of the correct transfer and blinatumomab, we get an increase in the number of cells that essentially die, and this is not the case in the presence of a bispecific control or a negative control construct. So thank you very much, particularly to the people that helped me do this project. And for any biologist interested, the QR code will lead you to my preprint. Thank you.